Very good afternoon to you all, and a warm welcome to the Golden Jubilee online lecture series, lecture number 3, प्रतिरक्षा स्वर्ण महोत्सव व्याख्यान नंबर 3। और आज के हमारे स्पीकर none other than Professor Narinder Kumar Mehra and हमारी चेयरपर्सन है Professor Shubhda Chiplunker. I'll reiterate this in English. Our chairperson, Professor Shubhda Chiplunker, and our speaker of the day is Professor Narinder Kumar Mehra, and we welcome you all. And we will start with introduction to our chairperson, Professor Chiplunker. Sushant, kindly display the slide. Sushant, slide लगा दीजिएगा डॉक्टर चिपलुंकर का. Thank you. Thank you so much. Full screen कर दीजिए. It's an honor and a privilege to introduce my mentor, Professor Shubhda Chiplunker, former Director, ACTREC, Tata Memorial Center, President, Immuno-Oncology Society of India, Education Committee member of IUIS, recipient of Shri R.J. Kinariwala Research Award and IIS Senior Scientist Oration Award 2007. Dr. Chiplunker had done her postdoctoral fellowship at Max Planck Institute for Immunology in Germany, and with none other than the renowned immunologist, Professor Stephen Kaufman. And the thesis was entitled Immunological Studies on Exogenous and Endogenous Murine Mammary Tumor Virus. She has been the former head of the Tumor Immunology and Immunotherapy Division at the ACTREC, and she has been a professor at Homi Bhabha National Institute. Her research has been focused on understanding immune dysfunctions in cancer patients and developing immunotherapeutic approaches for cancer treatment. So she started on this theme very early on when we hadn't even heard about the immune checkpoint inhibitor therapies. Her lab has contributed extensively to understand the role of gamma delta T cells, a special kind of T cells in solid tumors and leukemias. With more than 100 peer-reviewed research publications and 18 book chapters, she has been cited by Indian Academy of Sciences in a book on women scientists of India called Leelavati's Daughters, and the chapter for her is entitled Believing in Myself. May I welcome Professor Chiplunker and request her to chair the session. Thank you, ma'am.
Thank you, Dr. Taruna, for the kind words. Good afternoon, all. It is my pleasure to introduce a friend and a colleague for many years, whom I've known for many years, Professor Narinder Mehra. Dr. Mehra's name has become synonymous with HLA and transplantation and immunogenetics. His vast experience in the field and his contribution to the field of immunogenetics and transplantation justifies him. I would like to call him the HLA man of India. He was instrumental in setting up the histocompatibility lab at AIIMS, All India Institute of Medical Sciences, after he came back from his postdoctoral studies, and developing it into a reference center as well as a core lab for HLA research. He is credited with establishing the first Asian Indian donor marrow registry, which is a database of bone marrow donors. It would be really interesting to understand his journey in the field and his outstanding achievements. And I would briefly summarize his major achievements. The slide has already been put up, but I would like to highlight his journey, his achievements. He is currently the Vice President of the Indian National Science Academy International Affairs. He is the Fellow of the Royal College of Physicians, London, which is really prestigious. Recipient of several awards, among those are the Shanti Swarup Bhatnagar Award, Prize for Science and Technology. He has received the French honor, the Chevalier of the National Order of Merit from François Mitterrand. He's the recipient of the H.J. Mehta Gold Medal, ICMR Shakuntala Amirchand Prize, Sher-e-Kashmir Sheikh Mohammed Abdullah Award, Ranbaxy Science Foundation Award, ICMR Jalma Trust Foundation Award, and the list is endless. He has rich experience both in academics and in the clinics because he's been the former Dean of Research at All India Institute of Medical Sciences and ICMR's Dr. C.G. Pandit National Chair. He is elected Fellow of the International Medical Science Academy, the World Academy of Sciences, Indian National Science Academy, and National Academy of Sciences, India. He has been an active member and founder Secretary General of Federation of Immunological Societies of Asia Oceania, the FIMSA. He's been the council member of International Union of Immunological Societies. And he's organized several courses, advanced courses, is deeply interested in promoting education in immunology and training young immunologists. So he's organized several courses through the FIMSA and the IUIS. He's been the president of the Indian Society of Histocompatibility and Immunogenetics, also been the past president of the Indian Immunology Society. He's on several national advisory boards, being the one being the stem cell science, the advisory board of Indus Foundation, on the advisory board of INSA, and a trustee of the board of Immunology Foundation. I can go on with his achievements. He's a wonderful person, a great human being, always encouraging young people and young minds. With these words, I would like to welcome Dr. Mehra to deliver the Golden Jubilee lecture series, and he's going to talk to us today on HLA diversity and precision medicine. Dr. Mehra.
Thank you very much, Shubha, for those very kind words. Let me first try and share my slides, and then I get back to you. Can you share? Can you see my slide?
No.
This is trying to probably do that.
Yes, now we can see.
Can you see it? It's okay?
Yes.
Are you people able to see my slides, and can you hear me?
Yes, we can see and
Yes, sir.
And
hear
here,
you
thank
as
you
well.
so much. And I, at the very outset, want to thank Shubha for those very kind words. And I also want to congratulate the Indian Immunology Society for starting this series of Golden Jubilee lectures in the Golden Jubilee year. I think that this is a very apt thing to do. And we're already in the third lecture. We'll come on the 12th, and then the Congress will take place. And the organizers, please wake up and make sure that you do a good job next year, because after all, it's going to be a Golden Jubilee conference. What I thought I will talk to you today, I'll take you a little bit through my journey. When I show you this slide, it gives me nostalgic feelings of when I joined the Institute way back in July 1969. And I joined, and whatever I've learned in science is all due to this institute. Probably when I came in 1969, I didn't even know the spellings of science. And whatever I've learned is all due to this. This institute gave me an opportunity to work. This institute gave me an opportunity to interact with several people nationally and internationally, and made me wiser in my own field. So I chose my own field, and I thought I will talk to you a little bit of my journey, and but highlight on the HLA diversity and the precision medicine. This is where I started. It was in the 70s, early 70s, as a PhD student. We were working on immunology of leprosy. By the way, you know that in the early 70s, leprosy was prevalent. We used to talk about 10 to 12 million cases of leprosy around the world, of which 5 to 7 million exist in India. And we were trying to understand the immunology of leprosy through this mouse model. So much so that the CBA mouse became my friend, and I did hundreds of thymectomies, literally hundreds of thymectomies, total body radiation, bone marrow reconstitution, make the mouse immunocompromised, and do the M. leprae inoculation. We were trying to understand the host immunity in this mouse. But at this point of time in 1974, we had the opportunity to visit Varda. Varda, as you know, is a city very near to Nagpur, and there was a very famous Gandhi Memorial Leprosy Foundation there, and I had the opportunity to visit some villages there, and in the villages, the leprosy was rampant in that part of the country. Almost every family had a case of leprosy. But what struck us most was those large families of 5 to 7 children, only about one or two would be suffering from leprosy, others were healthy, despite the fact that they were living in the same environment, sharing the same room, eating the same food and all. And that was the time when Ron Nagal and Peter Doherty, you know that their papers came up in 1975 on the MSc restriction. And Benasraf had already indicated the concept of the IR genes. So we were just we were just trying to think about, is there a role for immune response genes in leprosy as well, because of the kind of families we saw in Varda. And that took me, and that I can say that the HLA bug then hit me in the early, and became my passion for life. But I had no equipment. These were my only tools that I had in 1974, the good old monocular microscope. In fact, my international friends even now sort of talk to me, how did you do this study through this monocular microscope? I used to use this microtest tray and shake it and therefore look at the cytotoxicity from the top rather than you need to look at from below with the phase contrast microscope. But this helped me to acquire my microscope. This was the first, this is probably in 1979. I don't remember. Maybe that's the time when I acquired the phase contrast microscope. And my group then started to grow. This is one of the colleagues at that time. I think Uma will probably recognize the hands. But my first paper came up in Tissue Antigens in 1975, the volume 5. This was the topmost journal of HLA at that time. Now, so therefore, this opened chapter, and today we have such a huge lot of diversity known in this system. We must pay tribute to these two gentlemen here, Jan Dosse from France and Jan Van Root from Holland, both of whom worked extensively at that time. Dosse on the multi-transfused individuals and Jan Van Root on the multi-parous women, and then were able to collect sera, the monoclonal sera for us, and we could do HLA typing in the 70s and 80s when I was doing the clinical service for selection of donor for patients requiring organ transplantation. We only knew about 20 to 50 alleles. And today in 2022, we know more than 35,000 alleles. And thanks to the DNA-based tools, again, we have to thank Carrie Mullis in 1986, who discovered the whole concept of the polymer chain reaction. And the 90s decade was a decade of technology explosion. The DNA-based tools became available to us. And now through the NGS, we have about 34 to 35,000 alleles already known. And we don't know yet whether we have come to the limit or not. The population genetics worldwide still remains one of the most important topics of the international histocompatibility workshops. So I think we will probably keep on finding new alleles and newer alleles there. But when you look at the HLA, immunogenetics, and medicine, in 20th century, of course, the focus was on transplantation, because the HLA system was defined primarily as a need for donor selection, organ transplantation, and then of course also from the point of view of his hematopoietic stem cell transplantation. And in 1973, when Bruton for the first time showed in UK a strong association of HLA-B27 with spondylitis, that the whole field grew of HLA disease associations there. A lot of studies on autoimmune diseases, infections and all. Technology update. The 21st century, on the other hand, we are now moving towards HLA medicine. Apart from the diseases I said, there is a lot to learn from schizophrenia, bipolar disorder, Parkinson's, and all. And there is this field of immuno and pharmacogenetics. The drug, who should get what drug. And I think the best example is that of abacavir. Abacavir is a drug which is very commonly given to patients with HIV, not as much in India as it is outside. And it's become clear now that patients carrying HLA B5701 should never be given this drug because they will immediately develop hypersensitivity reaction and many of them die as well. And similar is the case for many more drugs. I think this is the other field that is growing now, immuno and pharmacogenetics there. Regenerative medicine, of course, has come to play in the role of stem cells and therapeutics. Nanomedicine boosting the immune system for cancer therapy, and then the HLN cancer there. All of this is taking us to the whole field of precision medicine there. I have divided my talk into 2 or 3 sections, and the very first section is on HLA diversity in the Indian population. And we were struck with the amount of diversity that we have in the population of India. Indian subcontinent and the population diversity, probably the largest around the world. It's a unique gene pool, genetic linkages there. We have extreme MSC diversity in India. And our group has defined a number of novel HLA alleles and unique haplotypes. Many of them are disease associated. And I will, of course, in my talk, talk to you a little bit more about our studies that we did on type 1 diabetes. Of course, there is also a very important role on monogenic diabetes, which is becoming actually the subject of choice now. Pulmonary TB as well, and the HLA diversity and organ transplantation, because AMR still remains our problem. So towards the end of the talk, I will have a little time towards the HLA diversity and organ transplantation. They mimic a polymorphism and graft function, and of course biomarkers for early diagnosis of graft rejection. If we look at India, a vast genetic resource, large population, extended families, genetic diversity, 130 billion population, 28,000 endogamous groups. When I show these slides to my international friends, they get surprised with the number of endogamous groups that we have in India. Indian bank notes itself have so many languages now. And this article came in just last month indicating that the world population has already become 800 crores or 8 billion. And maybe by the 2100, it will become 10.4 billion there. So the diversity will keep on increasing. This is what has happened to the Indian subcontinent. I will spend very little time on this slide. One can spend 1 hour on this slide, but my purpose over here is just to highlight the point that the Indian subcontinent has migrations from almost every corner of the world. Indian subcontinent became like a melting pot of various racial groups that came here, intermingled here, and that would be one of the reasons for those several novel alleles that we have. So the extreme MSC diversity in India, and I wrote in the last, the below the line, Indian genetic diversity is second only to that of Africa. A lot of novel alleles, whether they have come up from the racial admixture, there is evidence for that, or selective environmental and microbial load. And the microbial load has been one of the singular factors driving this MSC diversity, because you know that the major biological function of HLA or the MSC is presenting the peptide, presenting the exogenous antigen to the T cell. And if you are surrounded by that many microbial antigens over there, you need a system. You need a very diverse HLA system as well. Our studies also indicated that the number of alleles that we find in India, several of them have origin also due to Oriental influence. And that was something new to us, because the typical Punjabi families in North India, we found the genes that exclusively are found in the Japanese and Chinese. Of course, we found a number of unique HLA haplotypes. Many of them are disease associated. And all this gives you a repertoire of peptide binding molecules. And of course, a good tool for you to work on vaccines as well. To highlight the point of diversity, let me talk to you about the HLA-A2. This is an antigen. This is a sort of molecule that is found ubiquitously around the world. Almost every ethnic group would have, and very large numbers. If I test 100 people in India, about 35 to 40 would have HLA-A2, but A2 has now been split into many molecular subtypes. Probably the number has gone beyond 400. But in the Caucasians, it is only one single type, the most predominant type, the O201, the yellow box. And in the Gambian and other black races over there, there could be a couple of them more, some of them which are uniquely found in that population, the O202 and the O205. Again, you can see the Japanese, the O201 is there, but then the other subtypes, the 206 and 207, which are exclusively found in the Japanese. And how about in India? And this is a slide I made many years ago in 2001, that we have almost everybody, we have a khichdi, we have the heterogeneity of this HLA-A2 is the largest, and that gives you example of how much diversity we have in the population of India. We have also these genes, the 207 that is exclusively found in the Japanese. We have the 205 that is found in the Jambians. And we have this red one, the O211. This red one is the one that is the unique, the novel subtype, only found in India. At that time, our papers indicated it is a special novel allele in the Indian subcontinent. We wrote in our studies until I went to Saudi Arabia a few years ago and I found it there as well. So maybe from that point onwards, you start to see this novel alleles there. But mind it, nearly one third of the HLA-A2 positive people have this novel subtype. And this novel subtype was also found in the Hungarian gypsies. I had the study done with the Hungarian colleagues, and we were collecting gypsy DNA samples there. And lo and behold, you find nearly 20% of A2 positive individuals in Hungary and in Spain had this subtype, fortifying the belief that gypsies anywhere in the world, around the world, must have gone from India. So I think there's a direct connection between gypsies and this novel alleles. A211 differs from O201 at position 73, 74 in the peptide binding pocket C of the peptide binding molecule of class 1 over there. And this is a very special subtype, as the aspartic acid at 74 is the most acidic subtype and all, but very close to HLA-O201, yet very different there. Similar was the case with HLA-B27. B27, as you know, is very strongly associated with spondyloarthropathies there, not only just the ankylosing spondylitis, but the whole spectrum of spondylitis that we know of. And in the Caucasians, it is one single subtype most commonly found is the B2705. B2705 also occurs in blacks. They have the other subtype, the B2703, which is a little bit protective amongst the blacks in terms of developing spondylitis. Azores is the only population that we knew at that time was really very polymorphic. And on the other side, on the eastern side, you have the B2704. 705 has gone down. Thai has the 2704, and they had the other subtype, the B2706. In the Indian population, you see that we have both. We have the B2705 like in the West, we have the B2704 like we have in the Japanese and Chinese, and we have the B2707, indicating the Indian subcontinent is the intermediary zone between the Western world on one side and on the East you have. So we have probably all subtypes, all alleles that the world population would have, and probably Indian subcontinent would be the best sort of place for establishing the donor marrow registries for unrelated marrow transplantation there. And this was also true when the whole study was done on NGS. The studies that I've shown you is not on NGS. Those were the molecular tools, of course. But NGS has also shown that nearly 1/3 of our HLA-A2 positive individuals are O211, carry that novel allele, which definitely does not occur in the Caucasians. And it may actually be found in other populations, definitely not in the Japanese and Chinese. It is in this region over here that you find this. And this is just one single example that I'm showing. There are many more examples of this. Similar was the case with the B27 as well. So the studies that we did 20 years ago have now been confirmed also with the newer tools of NGS as well. And I thought that the polymorphism plays a very important role. As the pandemic came in, I started to write articles for the newspapers on the pandemic. And my first article came, ironically on 1st April 2020. And the title was, Can India Be an Outlier in the Spread of COVID-19? And I had reasoned three factors which will be responsible for the observed low severity of infection in India. And when I say low severity, low numbers, or fewer deaths, this is in comparison to the Caucasian population. And the three factors that we talk about are the broad-based immunity due to the high microbial load, the Indian population, environmental factors and food habits. And of course, there is a whole lot of data in our Ayurveda over there that the food that we take, the haldi that we put, the masalas that we use, they are all immunostimulants there. And the possible role of genes that control our immune response, and here the HLA. And I showed you the extensive diversity of HLA in the Indian population with the existence of several novel genes and unique haplotypes. So our diversity of HLA makes it more possible for the race to present novel and new antigens, the new antigens to the immune system there. And when we look at the HLA-GWAS now, the Genomic Association database identifies 884 at the moment unique loci within the 4MB MSC, which are associated with at least 479 unique traits. The number of diseases that are now shown to be associated, some very strongly, of course, and the ones that we are highlighted are the ones are under investigation now. There's a lot of interest in HLA and cancer. There's a lot of interest, of course, in diabetes and infections, hepatitis, and other syndromes over there. So I will now switch gears and tell you the studies that we were doing for many years, for about 2 to 3 decades, on the genetics of autoimmune diseases in general, but on type 1 diabetes in particular. This word map tells you the incidence of type 1 diabetes worldwide. You can see there are high areas, intermediate areas, and the low areas. Ours is a low area. And then the African subcontinent, where the prevalence of type 1 diabetes is rather low. And this actually gives credence to the hygiene hypothesis that was put forward by Jean-François Bach from Paris, indicating the hygiene hypothesis. Because if you look at the Scandinavian countries over there, children are being born in Finland at 6 months of age or less than 6 months of age, diagnosed with type 1 diabetes. So the more the hygiene, the more is the autoimmune diseases over there, and if the infections and in the presence of infections, the autoimmunity goes down. We in India probably have a double trouble. We have both the infections and we have both the autoimmunity as well. Now, why is it that we have lower incidence of type 1 diabetes compared to the Western part, counterparts there? A lot of theories have been put forward. Exposure to maybe diabetes causing virus, we don't know. The only virus that we know people have studied is the Coxsackie B virus over there, or variations in vitamin D intake, and that could definitely be a very important factor. Vitamin D, because we are exposed to all this sun and all, and our studies on the Raika population of Bikaner area, which drink camel milk, also indicates that these people walk in the sun for long times, and therefore the incidence of diabetes in that population was found to be very, very low. There could be lifestyle changes, but more importantly, the role of genetics there. And this is where I will focus now. Give you an example of the population based differences in terms of genes that are susceptible to type 1 diabetes. When you look at the genetics of type 1 diabetes, of course, it is the autoreactive T cell mediated destruction of insulin secreting beta cells of the pancreas. More than 40 genetic loci are known to show strong association. Of these, five are very, very strong. Of these five, even HLA is the strongest, contributes about half the genetic risk. Of course, it is multifactorial. Prevalence varies from one part of the world to the other, from 0.1 in China to 37.4 in Finland there. Role of autoantibodies, these four autoantibodies are very important. There's also role of several non-HLA genes have been shown on this slide. And the majority of the studies that we have seen so far used to be in Caucasians. The Indian subcontinent, with its remarkable ethnic and cultural diversity, offers, of course, a unique opportunity to evaluate the impact of these genes in type 1 diabetes. And this is what we did. And I'll show you. And it was all a team effort of several of my colleagues, several of my students who then became colleagues. Your students then become your colleagues, and start to depend on them, several of them. So these are four major contributors of type 1 diabetes. If you look at from the immunology point of view, the peptides of proinsulin, or what you call autoantigens, are processed and presented by the APCs. They are shed from the beta cells of the pancreas, and they're taken up by the antigen presenting cells and presented to the CD4 T cell. Here, therefore, the diversity of HLA class 2, the insulin gene, the CTLA4, of course, is very important. And studies have been done by my other student, Neeraj Kumar, on that as well. PTP N22, we were able to develop a lot of collaboration with our Japanese friends, and a very important studies came out of that. And all of this leads to the generation of autoreactive clones of CD4 T cells which become activated and stimulate these CD8 T cells, which again go to the beta cells, peptides. So this cycle goes on and on. And here comes the very important role of these four genes there. This is a very important slide. I wanted to show you the genetics of type 1 diabetes and look at the differences that we have from Caucasians. In Caucasians, predisposing haplotypes, and I would now, of course, the terminology have changed, the designations have changed, but for the sake of simplicity, I will keep talking DR3DQ2 and DR4DQ8 because that's the molecule we know of. It's very easy to sort of talk about, although the nomenclature has changed. Almost 90% or more than 90% of children with type 1 diabetes in the West have either HLA-DR3-DQ2 or DR4-DQ8 or both. In fact, the relative risk, the synergistic effect of these two is even higher. In Indians, on the other hand, there is no effect of DR4 DQ8. Our susceptibility is only due to DR3 DQ2, and I will dwell on that a bit more. If we look at the protection gene, and we knew it for several years, that people who have HLA-DR3 but also have the DR2 DQ1, the protection gene is probably stronger. And this is the new nomenclature for that, for the DR2 DQ1, 1501 in the DRB1, 0102, and 0602. This is a dominant protection even in the presence of DR3 DQ2. In the Indian population, on the other hand, we don't have this haplotype. There are changes, as you can see here. We have the 1501, we have the 0103 rather than the 0102, and we have the 0601 rather than the 0602. So these are studies that have been done over the years, and we could find out the differences there. Again, in the Caucasians, the DR4 subtypes and what you call the O3, O2 haplotypes, not in Indians. And out of the several DR4 subtypes, the ones that are associated with type 1 diabetes in the West was found to be 0401, 402, and 405. In India, not, because of this 403. 403 is the most predominant subtype we encountered in the population of India. And this 403 is a protection conferring gene. This is proven beyond doubt, actually. And nearly half of our DR4 individuals in the population have the 403. And that could explain why there is no association with DR4, and also explains a sort of mystery that we had for many years on the other disease, rheumatoid arthritis, for example, where we had shown, and our rheumatology friends used to say all the time that in terms of number of joints and severity of joints, the severity of RA in India is lower than the West. And that could also be due to the fact that and rheumatoid arthritis, of course, associated with DR4. And maybe because of the O403 being the predominant subtype in India, our severity of RA is lower in India. And then, of course, we look at the combination of islet cell autoantibodies. The three autoantibodies are very important here: the GAT65, IA2, and the IAA. All patients with type 1 diabetes would have those. And depending on whether the child has one autoantibody or two antibody or autoantibodies or three, and that increases the risk of developing diabetes on one hand, but also if you can find out those susceptibility genes. And now I go to those susceptibility genes. The very important difference we encountered, and that was remarkable, three sets of HLA-B-DR3 haplotypes modulate susceptibility to type 1 diabetes in India, as against only one in Caucasians. In Caucasians, it is only the B8DR3, the red box. We have these two other boxes, the green and the blue, the B58 DDR3 and the B50 DDR3 over there. These were in addition to the three boxes. That was the first sort of difference we encountered there. But that was not enough. And when you look at the word map and look at those haplotypes there, every Caucasian population had the B8DR3 in the North America, Europe, Australia, New Zealand, anywhere, and the DR4 as well. So a combination of the two. And as you go to Spain, you get to see other as well, the B15DR3 now, not the B8 alone. In the Indian subcontinent, we have these three, as I showed you. B8DR3, like everybody has. B50DR3 was unique to India. We haven't seen this haplotype in type 1 diabetes in other populations. 58DR3 is what we see in China as well. Again, the intermediary zone. India is in the intermediary zone there. And what was most striking now, multiple DR3 haplotypes confer susceptibility to type 1 diabetes in the Indian population, as is seen here, the B8DR3, 58DR3, 50DR3. We then dissected this B8DR3 even more, and we were surprised to see this B8DR3, whereas in the Caucasians it is only single haplotype, is the HLA-A1 B8DR3, and they call it as ancestral haplotype 8.1. Ancestral may passing on from one generation to the other, and therefore be a cause of autoimmunity there. In Indian population, we had several. And the most common one was the B826 B8DR3. We called it as ancestral haplotype 8.2. And then we said, there's 8.3, there's 8.4, there's 8.5. The diversity of DR3 extended haplotypes that we found in India was much more than anywhere in the world. In fact, we also had this A1B at DR3, and we started to call it as A 8.1 variant form. And I'll talk to you what we mean by variant form. So the question in front of us was, is there a genetic advantage that we have? There's so many haplotypes, making you susceptible to type 1 diabetes. Does it signify disease heterogeneity? And our colleagues, all of them, Gurvinder, Uma, Neeraj, Gaurav, all of them created this whole set of microsatellites and SNPs and complotypes looking at genes between the HLA class 1, between HLA-B and the DR. There's whole lot of genes called the HLA class 3, which in the past used to be called third class genes, but they are not third class genes. They're very important because they also have the heat shock protein genes, the BAT genes, the TNF alpha, and things of that sort. So all of this was studied. And lo and behold, what we find, this is the A8.1 ancestral haplotype, AH8.1 in the Caucasians. Ours is the variant form, and I will show you why do we call it a variant form. Both look similar in HLA-A locus, the B locus, and the DR. Look at in the C locus, there's 701, we have 702. Everywhere else in the class 3 genes, our 8.1 is very different there. But what was the most striking was in the complotypes, the property factor, the C4A and the C4B, the complement types. And in the 8.1, there's a C4A null allele, and we don't have the null allele here. So even the other diverse forms of haplotypes that we had, which were associated with type 1 diabetes, they followed the same pattern like the 8.1 variant form. So we are very different from the Caucasian in terms of the susceptibility haplotype. So nobody had done this, extended haplotype studies, shown the differences there that so very well. And then came the International Consortium of Diabetes. They wanted to study, is there any functional role of the DRB3 gene? Because in their haplotype, each one of them is O101. We have the O202. And their study indicated that patients who carried type 1 diabetes, patients who carried O202 had a more severe form of type 1 diabetes. Look at in the Caucasians, they have this O301 and the DRB3 O101, red kind of a thing over there. And in the Native Americans, they tend to see then the other subtype, the O301, which goes with O202 in the DRB3 gene. Whereas in India, it is all the same. O301, O202. Our DRB3 gene is just singular one. This is our haplotype, type 1 diabetes haplotype. You don't do the extended haplotype. You only do the DRB1 and the BB3. You find it always here in India and not in there. So the severity influence of DRB3 gene probably in Caucasians, but not in Indians. We have not seen any influence of DRB3 O2O2 gene on the severity of type 1 diabetes. So there are subtle differences that modulate the risk of type 1 diabetes. This is the DQ constant. This is what they find in the West. They have the predisposing gene as the DR4 DDQ8, DR3 DDQ2, and then the protective gene over there, and just single. Of course, some DR4 subtypes, and like are shown over here, 401, 402, 404, 405, they were predisposing. 403 is predominant subtype in Indians, and it was found in the Caucasians also to be a protective gene. Hence, we have had no effect of DR4, the DQ8 on the susceptibility index of type 1 diabetes. And it is all DR3, where I showed you three haplotypes, three major haplotypes, the B at DR3, B50 at DR3, and the other one over there. So, and in this, of course, there were many more. The diversity of B at DR3 was very high. That was something very, very surprising to us, and we wanted to understand why is it happening. And of course I wanted to show you that in the NGS typing also showed to you that the 403 is the most common, is the most predominant subtype uh, in the Indians. This kind of a data, if you have side by side the data from the West, you can immediately say, this is from India, this is from France, this is from so and so, because they won't have that high O403. And that may be the reason that they're finding association also with DR4 and the severity of RA in that population is higher. We try to look into the hypothetical model for the origin and evolution of this autoimmunity favoring DR3 positive haplotypes. And of course, anybody will think, okay, there could be a common ancestor, the DR3 DDQ2, and leads to 8.1 in the Caucasian, 8.1 variant form, perhaps in the Asian Indians, And from there you have all those multiple subtypes formed, multiple haplotypes formed. The other way could be, well, they have the 8.1, we have the 8.2, because 8.2 is the most common susceptibility haplotype in the Indian population. In fact, 8.2 should be 8.1 and 8.1 should be 8.2, just because they encountered it first time. So just because they discovered it earlier than us, they called it 8.1. And we believe that the common ancestor, the ancestor must be 8.2. From there you have the 8.1. We have the 8.1 variant form, and we have got many more. The diversity is more in the Indian population because the Indian population probably had greater opportunity, greater time for environmental stimuli there. And by way of joke, I do tell my Caucasian friends that you need to be born 1,000 years down the line and you will find your 8.1 has also become diversified. Diversity happens over time, over exposure to environmental stimuli. So the next part I want to talk to you very briefly about TB as well, the pulmonary TB. And we did this study in the Saharia population in MP. Saharia population where the incidence rate of TB in this tribe has been very high, 95.6 cases per thousand a year. High morbidity, high mortality. And a lot of people, the geneticists believe that they have kinship with the Munda language. And they could be Indo-Aryan speakers linguistically. Of course, it's a tribal group, very close to Bhopal. And we did this study over here, and our question was, why is it that they're finding very high incidence of TB in this population? Is there a genetic influence? Of course, by the time we had already shown that this particular haplotype, A2B40 DRB115, which is a DR2 haplotype, is associated with leprosy and with TB both. And this haplotype, you can see, lo and behold, you find very strong, very high in the population of Saharias. And if you compare that with the neighboring population, Bhil is another tribe over there. And the incidence of tuberculosis in the two of them differ so very significantly. Whereas TB is very high in the Saharias, maybe this could also be a factor and not as much as in Bhils. Let me to the third part of my talk, focus to kidney transplantation. Uh, precision medicine in kidney transplantation. After all, the HLA system was defined primarily because of need in organ transplantation. The role of donor-specific anti-HLA antibodies, we have shown it a number of times. Several papers actually came out from our group as well, associating these antibodies with the antibody mediated rejection. But I want to focus today on the MICA. This is another very strongly polymorphic genetic system, lies very close to HLA-B locus over there. And there is a role of this MIC polymorphism, MICA antibodies and graft function as well. And then I'll talk to you, if the time permits, utilizing biomarkers for early diagnosis of rejection. The issue here is there is a relentless tissue injury going on in clinically stable, the so-called clinically stable individuals who have had a kidney graft leading to subclinical injury to the graft, which cannot be identified otherwise, and that is poorly understood. And this is often missed by the histology, and maybe we need to develop some biomarkers. So the whole field is actually moving towards those directions now. So there are issues and concerns in solid organ transplantation are two. One is a good HLA matching. Good HLA matching leads to lower grades of sensitization, decreased requirements for immunosuppression, lower risk of de novo development of donor-specific antibodies, and thus decreased episodes of AMR. HLA antibodies, of course, are the cause of the chronic graft failure in renal transplantation. And the antibodies that cause ABMR are mainly made by the memory B cells. warranting establishment of in vitro B cell assays in which the memory B cells are selectively differentiated into plasma cells and are used to make antibodies. Unfortunately, unlike the T cell assays that have long been used to control T cell mediated rejection, the primary cultures of B cells are not very easy to maintain. So therefore, the interpretation today of the current donor-specific antibodies results is challenging and includes several factors over there. And I don't want to go into the details of that. Several studies, several groups have already indicated that presence of HLA-specific donor-specific antibodies are the cause of the graft loss. And there is a vicious cycle of HLA mismatched transplants and ABMR. Oh my God, it keeps fixing. And look at this patient here. A patient in the middle, over here, kidney failure there, waiting on dialysis. First transplant is done. That's the time this guy is non-sensitized. You give him a donor recipient HLA mismatched kidney, leads to the development of de novo HLA DSA, leads to AMR, kidney transplant failure, again comes back on dialysis, needs a second transplant, a repeat transplant is done. But by this time, this guy is already sensitized because there are already antibodies now there. And there is increased risk of HLA DSA, again leads to AMR, kidney transplant failure, and needs a third transplant now. And by the time is highly sensitized, would lead to AMR, permanent dialysis, back on dialysis, and even death. What do we do? These are the patient impacts over there in terms of, but the solution is avoid donor recipient HLA mismatching. This is the crux of the whole issue in solid organ transplantation. Do HLA typing even for patient donors who are not from the within the family. Try and give a well matched donor recipient pair, or look for what we now have a sort of software for the permissible mismatches there, but then there is a need for regular screening for antibodies. Only then you can break this vicious cycle. I had these two books. One came up in 2010, The HLA Complex in Medicine and Biology, a resource book. And then this was followed by this book from the Frontiers, Through the Frontiers in Immunology on Antibody Repertoire and Graft Outcome. And I'll be happy to send these soft copies to anyone who would need it. I want to, rather than HLA antibodies, want to talk to you about our studies that our group did on non-HLA antibodies. Now, some of you will ask, what is the meaning of non-HLA? Now, non-HLA antibodies, antibody against donor endothelial proteins and biomarkers that indicates events like acute organ failure or injury. And the term non-HLA antibody is used in transplantation to describe autoreactive and alloreactive antibodies specific for targets other than HLA. And the commonly reported targets are angiotensin, the LG3, vimentin, tubulin, collagen, MICA. MICA is the most important one. MICA is the MSC class 1 chain related molecule A, very near to the HLA-B locus. It has now been described as the novel histocompatibility locus in solid organ transplantation. This is our MICA, close to HLA-B locus, expressed on natural killer cells, ligand for NKG2D, on gamma delta T cells and endothelial cells. And our group has been working on these three lines of evidence on make antibodies against the donor endothelial proteins leading to acute graft loss or failure or injury. A dimorphism is study that we have done because of lack of time, and I'm going to talk to you about that at 129 of the alpha 1 domain, describing people into strong or weak binders of NKG2D, the methionine or the villain there. And third, of course, is very important role with soluble MK levels as well in the graph outcome. These are the two students who now became friends and colleagues, have done these studies, make antibodies and renal graft outcome. We had several papers actually describing from our group, the impact of Mickey over there. I want to summarize only on this, this paper there, which came up in 2020, that Mickey is also like HLA, probably a novel histocompatibility locus in solid organ transplantation. And our allele typing, we have not only looked into the Mickey antibodies there, but also the Mickey alleles as well. And it indicated to us that like HLA, there could be donor-specific antibodies to Mickey as well, which would be able to sort of harm the graft. So this raises the question, therefore, should Mickey screening and Mickey allele typing be introduced in solid organ transplantation on a regular basis or not? I think there is a mixed reaction now. A lot of nations, nephrologists keep calling us that could you also do the MIC typing for us? I think slowly it is gelling in that we are losing grafts sometimes where there's no evidence of anti-HLA antibodies over there. The graft is still lost. And this could be due to the non-HLA antibodies and mainly to the MICA antibodies there. So I think more data will need to be established to make sure if this could become also like a very important tool in addition to HLA for the selection of a donor for solid organ transplantation. A study came up just this year from France, from Samik Barham's group, who also confirmed our finding that mimic gene is a histocompatibility antigen in kidney transplantation. He looked into the preformed and the post-transplant donor-specific antibodies and found that patients who had antibodies both to the MICA and to the HLA, they tended to lose the graft more uh, quickly, actually. So again, I think such kind of data will come, and then we need to then develop new technologies and tools. If we can just in one single test for both the HLA and the non-HLA and antibodies actually for the transplant. And the last part, everybody talks of the precision medicine in kidney transplantation. Is it just a hope? Is it just a hype? Or is it realistic hope? This is a good paper I could get hands on, which came up in Transplantation Direct into 2021. They've tried to summarize everything. We need to develop those pharmacogenetic panels, both for the recipient and the donor over there. The precision pharmacotherapy would lead to risk stratification and personalized immunosuppressive therapy. By the way, you know that patients who go for organ transplantation, they have to be given a very heavy doses of immunosuppression. And of course, one shoe size fits all is a thing of the past. And depending on the cytochrome 50 genes, as you can see here on this side of the slide, you can actually identify those who need lower doses of immunosuppressive drugs, those who need higher doses, those who are at higher risk of losing the graft and from the others. And this is a study being done by our colleagues in San Francisco there, where they're looking at the gene expression score through the transcriptome window in tissue, and they feel that they could predate the clinical and the histological injury. The question here is, are all histologically stable allografts? The surgeon does the biopsy and does, oh yeah, the kidney is doing fine. Is it truly clinically fine? And it could be molecularly unstable. And their data indicates that this is the zone, the yellow zone is the one which apparently looks like a stable graft, but when their gene expression score tends to grow more than 32, this is the group that could actually eventually move into the red zone over there, where the Q score becomes very high. These are anyway early days, and I'm sure, and this is a test that they have developed in urine there, looking at the gene expression score, and the take-home message from such studies is that the cell state transcriptional formation information could help define a clinically stable graft into histologically versus molecularly stable graft over there. And then looking at the score you could identify those who are undergoing what we call clinical stage of injury there, and therefore something can be done at that stage to save the graft. Well, friends, I think today we have moved into the era of translational research, which has become the need of the day, and everybody keeps talking about bedside to bench, bench to bedside. Scientist is in the middle of the whole thing over here. You're right here, and you have all these tools of genomics, proteomics, transgenics, knockouts, knock-ins, structural biology, and you name it. And you need to interact with the clinicians on the one side and keep doing the clinical trials of new tools and technologies that you develop in the laboratory, all for the prophylactic and the therapeutic solutions. And this is therefore my last slide, that we are now in the era of, my friends in genetics call it the era of genetic medicine. I call it the era of immunogenetic medicine, where the way forwards for us is connecting basic research to patient care, understanding the immunogenetic basis of the disease, developing specific genetic tests, identify those at risk of graft loss, and devise preventive and therapeutic treatments there. And in all of this, HLA has emerged as an important tool for personalized and precision medicine there. So our four P's: personalized, participatory, preemptive, and predictive. I think uh, in my own career of more than 5 decades working in the lab, and now, of course, as a mentor for several, we have learned so much down the line, and so much more needs to be learned. After all of us, even if you are working the mouse model there, we are ultimately concerned with the human health and not the mouse health. The mouse is just a model for you to develop new tools or to develop new understanding there. So towards the end, I want to acknowledge, I don't want to name each one of them, all my colleagues, students, associates, and many others. Your 50 years of your life, you meet hundreds and thousands of people, both within India as well as outside. Together, it has been a learning experience for me, along with them. And I want to also thank the several funding agencies who have always helped us with the funds. Thank you very much, Aruna, for giving me this opportunity to give this lecture in the Golden Jubilee year. Thank you very much.
Thank you, Narinder, for such a wonderful talk. So lucidly explaining your journey and making this complex topic so simple to understand. You have brought out the right point. We Indians, the diversity in we Indians make us very unique and different compared to the rest of the world. And that throws in many challenges also for treatment. There are some interesting questions, comments on the chat box. And the one from Dr. Aikwar is, sir, you have excited us enough to dig in and find out our own HLA types. There are
Yeah.
questions.
Yes, I remember in 19, I think 78 or something, I wrote an article for some magazine, and I gave a title, but the editor changed the title to "Genes Are Your Fortunes." And this is absolutely true. This particular field has shown you genes are your fortunes. Gone are the days when you must carry the teva of the boy and the girl and go to the pandit to sort of see if the stars meet or not. You need to do this kind of things, and I haven't, because the time was too short. In your life of this 50 years, you do many more things that you cannot talk 1 hour time. And I think the other very exciting field is that of thalassemia there. The thalassemia major is a big problem for India, and how we have been finding donors for them, also through the prenatal genetic testing, cord blood transplantation, things of that sort. So many more. Yes, the genes are very, very important. And here we are talking of genes that are directly involved with the immune response. So these are IR genes that we're talking about.
I just want to ask, are there, today we talk of checkpoint inhibitors, CAR T cells. So are there real good reports which have come out regarding prediction with HLA subtypes? Or
Yeah, so I said that yes, there is a lot of interest now in HLA and cancer. A lot of people are talking about it. Maybe at some point of time, one can give a lecture on HLA and cancer. There are, and immunotherapy is already on the horizon now with both the the CAR NK and the CAR T cells. In various diseases over there are companies that are making disease specific CAR NK cells and all. And here you want to find out, not everybody will get benefited from there. So I think the therapy benefits for those, there have to be biomarkers there, and there are studies being done in that direction. That's where the 21st century is really going about.
yeah, here again we Indians will differ because there's a report from Mary Carrington's group recently, very recent in Jan, which said that HLA-BO3 is the one which can predict response to checkpoint inhibitors in one of the malignancies.
Yes, and Mary, of course, has been a good friend. We have been collaborating with her for very, very many years, and she's, of course, looking into all of those things. And I think that this is where my younger colleagues should actually work on, because if COVID was not there, today we have been only talking about immunotherapy there. But those 2 or 3 years that have been lost will now be gained actually, and you are going to see more of it. You're going to see more news about that as well. And I think it's time that some of our colleagues start thinking in terms of looking into HLA and cancer.
That's right. I see some questions. One from Jyoti Kode is, these days mismatched transplant donors for allogenic stem cell transplant are gaining importance for improved clinical outcome. How do you read it for organ transplantation? I think you've explained it
Well,
already.
organ transplantation, we have been doing mismatched transplantations. There will also be 6 out of 6 mismatched transplants are done. After all, if when you take kidney from a cadaver donor or from the spousal transplants that we do, they don't match. Yet they're doing well, and therefore the whole concept of permissible and non permissible mismatch has come into play. The problem is for the hematopoietic stem cell transplantations, where we would love to do a HLA identical sibling transplant, but those are not they are to come by. I mean, only 1/4 of the children in the family would actually be HLA identical, and our own experience in AIMS has been that nearly 30% of the families that come to us for a bone marrow transplant would end up having a HLA identical SIB. Many of them will not have. Now the focus is more on haploidentical transplants. Haploidentical you can take from the mother and the father over there, and there of course also will come the role of the KIR typing. KIR is another, the killer immunoglobulin receptor genes over there, and mismatch of that. So I think still a lot needs to be learned about that. People have been also talking about the cord blood transplantations as well. We are creating now the national guidelines for cord blood banking, but cord blood is losing the steam for the thinking that the cord blood cells are not immunogenic as much as the others are. That's not the case. People are now moving more towards the haploidentical transplants, but the success rate is still compromised on those. The best is the matched transplants.
I see a question from Dr. Taruna. Are there HLA chips commercially available, or do you see there is a realistic possibility for such HLA chips that can type an individual's HLA profile and predict their susceptibility, protection to various infections, cancers, autoimmune disorders?
Well, yes and no. There are when you talk of chip, I'm sure you're talking of the HLA B27 kit or HLA DR3 kit and things of that sort. Yes, the B27 kit is definitely there, And most of the labs who do HLA typing, they end up doing about 100 patients typing a day, finding out whether this individual has the HLA B27 or not. But in addition to the B27, even the B27 negative individuals also need to be. So, you need to have a kit that has the B7. We can't just have the kit that is primarily developed for the Caucasian population. We have to have our own. But there are no specific kits for autoimmune diseases or for type 1 diabetes or rheumatoid arthritis. The other disease which shows a very strong association, the narcolepsy. The narcolepsy is the daytime sleepiness where one single haplotype is known to be so strongly associated. So yes, there is a need to develop this, and as the technology and tools have improved, NGS-based typing has come up. I'm sure the next step would be to have those kind of, you call them chips, or maybe the tools that you can use to identify whether this individual has the gene or not. Yeah.
Uh, I don't see any more questions, but I see a lot of appreciation messages coming in. Gaurav Sharma, Uma Kanga, Rian Rao, and so many more.
I can only say namaste to them with folded hands. I have, I just tried to show you how I started with a very humble beginning. Actually, I had nothing in the lab, I can tell you. And I remember I went in 1976 to Holland to Van Root's lab, and he offered a dinner talking to somebody. Here is this young man who's come from India. I see that he has not, because he had come to our lab and he saw, and he said, I can see that he has nothing in the lab, but he has the inspiration and he has the
Right.
desire to do. So this is a message from me to my young colleagues. Do not, don't lose heart. Just go on. And if you believe in something, just go on and on. There will be a lot of people who you will meet in life who will come forward to shake hands with you and take you around. This is what I have done. I started from zero, absolutely nothing. I showed you the microscope that I had. But then, your desire, your self belief, your confidence should never go down.
Right. Dr. Mehra, there are two questions, and I think we have to take those.
Yes.
One is from Shripat Patil. Professor Mehra compliments always. Uh, through HLA typing, we can trace the migration of population.
Yes.
Yes.
Yes. Sure. And I can tell you, of course, we did not chase that study. We should have. And that's a regret that I had. In the 80s, we had this serology as the tool, and we did some studies in the Indian population. And I also had some data from my colleagues in Australia who had raised the similar data in Polynesians and Melanesians on the Australian continent over there. And we found so many genes common. And a couple of ambassadors I met. There was the ambassador of India to Australia and the Australian ambassador. He said, "Oh my God, this is the kind of study we need, where you can show the kinship. That maybe the Polynesians and the Melanesians have actually all gone from India." I showed you that example of the gypsies of Hungary and Spain, and your grandmothers used to tell that gypsies anywhere around the world have actually migrated from India. This is direct proof. Look at that O211. In the predominantly Caucasian population, you find that only yes, population migration, it does help. So that therefore, the population genetics still remains one of the hot topics of the international histocompatibility workshops.
Take one last question, which I see on the screen. That's from Ramesh Rai. Sir, nice talk. Can you shed more light on new HLA nomenclature? How do you understand it in easy way?
There's no easy way. Well, it is easy. I would say that you must read history. History is very important. How did people define HLA A1 or A2? And after A1, A2, there is no, and there is A3, but there's no A4, and there is B5. and there is no A6. So how did this happen? So I think unless you know the history, once you are conversant with the history, it becomes easier for you to then follow that. Of course, now with the molecular tools, earlier we used to only call it as a HLA DR2, then we started calling it as a HLA DRB1 star O2, then O2O1, O2O2, and then now synonymous changes, non-synonymous changes. You go on, you go up to at least eight points after that, the colon there. So those are, and that is actually because the NGS is helping you to define the allele at the single nucleotide level. That's what you need actually for bone marrow transplantation. For the HSCT there. The example is that of the Nightingale twins, where the organ transplant was done between the twins, and that graft functioned for 51 years. If the graft could function that well in the monozygous twins, and you, with the current knowledge of immunology and with the tools in hand, you should be able to now find all those barriers to the transplantation there, break them all, and then make sure that the graft can go on and on.
Uh, I hand over to Taruna. Thank you, Narinder, for a wonderful talk. I always enjoy your talk.
Bala
Sir,
has a question. Bala
uh,
has always the last word.
actually, sir, there is one question from Dr. Teresa Mary from Chennai. You please look into that, sir.
Wait. It's gone up, I think. Can you read
Yeah,
the question if you have it in front of you?
is there any possibility of 100% match of donor and recipient? See, come across a case, it seems.
Yes, I mean, we do say theoretically the possibility that my HLA phenotypes will be similar to that of yours is 1 in a billion chance. But when you come to a particular population, Indian population, that figure will go down. And when you go to a particular ethnic group, there are so many ethnic groups in India, endogamous groups, yes, there is a chance. There is a chance if you're looking into an endogamous group, particularly in South India, you know that 1/3 of the population in South India is endogamous. So there, the possibility is higher, yes.
But sir, shall I ask you one question, sir?
Yes, why not?
I am Dr. Teres Mary. Just now you discussed, sir. Thank you for your wonderful lecture, sir. The thing I want to clarify is actually there will be inheritance of one from father and one from mother. Like that only we are being taught, no? Here in this particular case, the donor was father and the recipient was son, and both of them had similar A locus product, B locus
Yes.
product, and C locus product.
Yeah.
So
Yeah.
this is the first time I'm seeing like this. I have done almost
No.
500.
We have seen in the past. I have seen it in the past, and if you look into, you will find that the mother and the father probably are cousin marriages there. In the cousin. If not these two, but go back into the family tree, many of them in the past, this would be a close cousin marriages there. So by chance, an ancestral haplotype. There are many haplotypes that are also found. They have the linkage, the disequilibrium there. The A1 BA 3 like, or the A26 BA 3. It's just that possibility that the
Yes,
husband and the wife are sharing one haplotype with each other, and
yes.
they have actually transmitted the same onto
Okay.
the child, and therefore the child could also become,
Yes.
identical.
Okay. Thank you, sir.
Yeah.
Thank you.
Ma'am, there is one more question by Aishwarya Lakshmi M. Which exons on the complement factors C4b and C4a is critical for typing?
Oh, C4A and C4B is another very important gene. Nobody, people have not given too much of attention to it. As I told you, that C4A and C4B are both polymorphic there. In those days, when I was studying those, there were about 10 to 12 alleles of these known. I cannot say to you which particular exon that you have to really look into and find out. But all that I can say is that the C4a null allele that you find in the ancestor of haplotype 8.1, that A1, B8, DR3 that the Caucasians have, all of them have the null allele. The C4a is null, zero there. We don't have that null. And the studies at that time indicated that the null allele is a functional gene. It is also associated with 21 hydroxylase deficiency there. So one would have to do studies to indicate, to find how many patients of type 1 diabetes also have or would have in due course of time 21 hydroxylase deficiency there. Fortunately, we don't have, but our other haplotype, I think the B50DR3, that has the null allele, but the null allele is in the C4b and not in C4a. Yes, they're very important genes. So when you talk about complotype, you talk about C2 gene, propertin factor BF, C4a, and the C4b. That makes it a complotype, you know, and look into that in autoimmune diseases, and I'm sure you're going to find something very useful there.
Yeah.
Uh,
Over to you.
Thank you so much, ma'am. And now it is over to Dr. Balakrishnan. Sushant, kindly display the word of thanks slide. Thank you.
Yeah. Good evening, everyone. First and foremost, on behalf of the Executive Committee of Indian Immunology Society, with immense gratitude, we express sincere and wholehearted thanks to our eminent speaker, Senior Professor Dr. N.K. Mehra, INSA Vice President, for delivering the prestigious third IAS Golden Jubilee Lecture and sharing highly valuable data on HLA in clinical practice as well as in population genetics. Sir, it was really an exhaustive and interesting data sets for all of us. We are honored by your presence and sharing your rich experience in the field of clinical immunogenetics. As inspired by your talk, some really wanted to take their HLA. That shows the level with which you inspired them. Thank you very much, sir. We express our happiest thanks to chairperson of this event, Professor Subhadra Sipulkar, former director at Trek, Mumbai, for having accepted and nicely orchestrated this event in a scientific way. Thank you very much, madam. We thank all the participants across the country for their overwhelming response by joining and interacting with us. Thank you all. We place on record our sincere thanks to the IT team, ICMR, NARRCH, for their seamless support and work in making this event a grand success. Thank you, one and all. Thank you.
Thank you very much. Thank you. Bye-bye.
Thank you.
Bye, everyone, and we'll see you next month. Thank you.
Next
Thank
year.
you for such an interesting talk, sir.
Thank you very much.